Okay, our last topic of the semester is going to be cancer. Um, so, obviously, cancer is a cellular disease. It is a disease of an abnormal cell cycle, which leads to cancer cells proliferating uncontrollably, so they are no longer following the normal cell cycle. And then they gain the ability to metastasize via the lymph or the bloodstream and grow and develop in other parts of the body. So when we talk about cancer cells or tumor cells or things like that, the better terminology to use aside from a tumor is a neoplasm or a new growth, essentially. There is a difference between benign and malignant growths. Cancer is associated with malignant growths. Benign is a uncontrolled growth that is localized. It is not metastasizing, it is not spreading. That doesn't necessarily mean that it's not dangerous. If you have a benign tumor that is pressing on a major nerve or a major artery or a portion of your brain, then it's definitely not, without a problem. It's not cancer, but it's definitely still harmful. Malignant or cancer tumors or neoplasms are not only an abnormal growth, but also a spreading type of growth. They have the ability to move throughout the body and begin to grow in different areas. Cancer has many causes. In general, we can say, and we do say, that cancer is always a genetic disease. Now, what actually causes the change to the genes could be a spontaneous genetic mutation, or it could be an environmental cause, a carcinogen, a virus like HPV. So there's a multitude of different things that can induce genetic mutations that lead to cancer. When we look at cancer cells, we see that there are a number of different things that cause essentially genetic instability. Generally, our genes should be pretty stable going through the cell cycle, things like that. But we see in cancer cells that there is an inability to repair DNA issues, an inability to replicate DNA properly. Our checkpoints that we talked about in our cell cycle, they no longer are able to stop the cell when there is a problem. We have mistakes that happen in mitosis. We end up with abnormal chromosome numbers, abnormal chromosome arrangements. We get breaks in chromosomes. So there's a lot of different genetic issues that contribute to cancer. But cancer is always a genetic disease. The origins of cancer always lies in a change in the genetic code. Generally, it takes a lifetime to develop enough mutations to cause cancer. So when we look at the cancer incidence, you can see that it goes up dramatically the older you get. So after age 50, 60, it is a huge increase. So that's simply because it takes a long time to gain enough mutations to cause cancer. Cancer is not a one-hit wonder. It requires multiple genetic changes, and usually it takes a while to get there. Here what we're looking at is breast cancer. So this is a karyotype from breast cancer, and you can see that there are multiple translocations here. So these chromosomes should all be the same color. This should be a blue one. This one's two different blue. This is blue and red. This is green and pink. Blue and yellow. Green, pink, and a different pink. There are so many translocations in here. So that it takes a huge amount of genetic change to actually cause cancer. Now, when we talk about the fact that cancer is always a genetic disease, there's two different ways to get an issue or a problem, a mistake in the DNA. Germline would be an inherited mutation. So, in terms of hereditary cancers, you can inherit the mutation from one of your parental chromosomes. Most cancers are not germline. They are not inherited cancers. That's not to say that there is not possibly some predisposition involved, but when you look at cancer being a genetic disease, only about 10% of cancers fall into this germline or an inherited type of cancer. The rest of it is spontaneous. So in this case, spontaneous means that there was a point where the DNA was normal and it gained a mutation. So this could be a truly random mutation, or it could be a mutation that was driven by exposure to carcinogens, exposure to oncoviruses. But at one point, the DNA was normal. It was not inherited this way, but something has induced the change. Again, it's not a one mutation disease. So if you look at how this progresses, you've got a mutation that gives this cell an advantage. So any type of advantage means that it's going to allow that cell to have a better chance of surviving. So it survives. And then you get a second mutation that increases the overall advantage. A third mutation allows the invasive ability. Then you begin to see the spread and you begin to see cancer. So it takes a number of successive mutations that are not being repaired for cancer to develop. When we look at it, the current thought is that every gene that we have is going to have somewhere in the neighborhood of 10 to the 9th power mutations over our lifetime. We've got at least 25,000 genes, and each one of them is going to experience 10 to the 9th power mutations. So you kind of have to ask yourself, well, why don't we all have cancer? Number one, not all of them survive. So we have a mutation. We've got good P53. It stops the cell cycle at the G1S checkpoint. P21 is made, and we're able to pause the cell cycle in order to repair it. So that mutation, even though it happened, was not passed on. Maybe it was passed on, but it still takes a number of mutations in a row to get to cancer. So even though we do have a relatively scary high number of mutations that occur within our DNA over the course of a lifespan, we don't all have cancer all the time because we have the ability to repair this, and B, it takes a huge number of mutations to actually lose the ability to control the cell cycle completely. So if we look at cancer cells, they have a certain set of characteristics that make them sort of stand apart from the normal cell that we've kind of been talking about for the whole semester. So the first would be that everything we've talked about so far, remember the entire chapter on signaling, we had to tell cells, grow, 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 live, 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 live. We had to tell them everything that we wanted them to do. Cancer cells are not terribly dependent on growth signals, survival signals, division signals. they kind of just do this stuff independently of the signals that would be telling them to do it. Combine that with number 2, which would be a decreased rate in apoptosis. So what we learned in the signaling chapter is that we have to tell them to live, we have to tell them to grow, we have to tell them to divide, we have to tell them to differentiate, and if we take all the signals away, then they die. They don't really die. So, if we look at the checkpoint and P53 is supposed to stop the cell cycle. If we lose P53, we're not going to stop the cell cycle there, and we're certainly not going to trigger the cell death of a mutated cell. So they're no longer responding to the normal cues that are dictating when the cell should live, when the cell should die, when it should grow, etc. Third thing is they have the ability essentially to be immortal. They have an indefinite ability to proliferate. This is weird. Most cells have a mitotic clock in the form of what are called telomeres. These are non-coding sections of DNA at the ends of chromosomes, and every time the cell divides, the telomeres get shorter and shorter and shorter and shorter. When the telomeres are gone, the cell dies. It triggers apoptosis. And we've seen in the lab that cells know how many times they've divided. Doesn't matter if you move it to another colony, it's going to divide a set number of times, and then it's going to die via apoptosis. So cancer cells have an enzyme called telomerase, and it is essentially always turned on. Telomerase adds length to the telomeres. So instead of shortening with every cell cycle, they continue to lengthen, which means the mitotic clock is essentially gone. When we look at cancer cells, we see a huge amount of genetic instability and a huge number of mutations, just like we saw when we were looking at this breast cancer. We've got a lot of mutations, a lot of instability, a lot of translocations. There's no reason this cell should still be alive. There are so many genetic issues in there that the cell should have triggered death, but it didn't. We also then have an abnormal invasiveness. We just learned in our last lecture that cells are adhered to something, either the extracellular matrix or the basal lamina. Cancer cells begin to lack the proteins needed to adhere them to things. So, they don't have the cadherins that we need to actually attach to something else, which means that they have the ability to move when generally we don't want cells to move. They have completely abnormal metabolism. When a normal cell uses nutrients, mostly the nutrients are going to be utilized to make ATP. So we're going to utilize in oxidative phosphorylation, we want to make ATP. These cells don't care about ATP. All they want to do is grow and divide. So they just want to go through the cell cycle as fast as possible. They have no interest in generating energy. They simply want to grow and divide. And then the last thing is that these cells, not only do they gain the ability to invade here, they can actually survive and proliferate in a foreign tissue, which is completely abnormal. Most of the time, if a cell finds itself in an environment that it doesn't belong in, it triggers apoptosis. And they don't. So when a breast cell finds itself in the lymph, and then in the bloodstream, and then in the liver, it somehow survives and proliferates there instead of dying. So you can kind of see that all of these put together is going to create a bad situation for a cell, because we're no longer going to be able to control it, and it's not going to act like a normal cell would. So there's two different types of genes that are involved in cancer. The first is what's called a proto-oncogene, and the second is what's called a tumor suppressor gene. Proto-oncogenes are going to undergo what are called gain-of-function mutations. The normal role of a proto-oncogene is to drive the cell cycle. If you want to think about the cell cycle as kind of the gas pedal and the brakes in order to properly grow and divide, proto-oncogenes would be pushing down the gas pedal. So they are supposed to drive the cell cycle, which is a totally normal thing that we need to happen. What happens when you get a mutation in a proto-oncogene is that it's like slamming your foot down on the gas and not being able to pick it back up. It's going to lead to excess cell survival and proliferation. So we're going to grow and divide well beyond what we should. Now, proto-oncogenes are also what's known as a dominant mutation, in that you only need the mutation in one of the copies to create the excess growth. The other type of gene involved is a tumor suppressor gene. The normal function here is going to be the brakes on the cell cycle, to say, oh wait, nope, we need to hold off. So like P53 that we've talked about before. These are loss of function. So when you get a mutation that affects both, that's why these are called recessive mutations, because you actually have to have the mutation in both of the copies, you're incapable of pumping the brakes on the cell cycle. So it again leads to excess cell and survival and growth. So it doesn't matter which one is involved, you get the same thing, and in reality, both are involved. So if we look at the types of gain-of-function mutations, what do we see? How does this work? Depends on what happens. If we get a mutation in a coding sequence, so within the actual gene, generally we see that we end up with normal amounts of the protein, but it's hyperactive. So it's going to be driving the cell cycle too much, utilizing a normal amount of them. If we get gene amplification, where we actually see the gene being multiplied over and over and over again, then we're going to see that the protein is normal, but there's too much of it, so you end up overactivating the cell cycle. Third thing could be a rearrangement in the chromosomes here. So if we get an issue with a nearby regulatory gene, we can start overproducing the normal protein, which again is going to lead to overactivation. If we get this proto-oncogene fused to a normally active gene, then you end up with a hyperactive. So you can either end up with the protein in normal amounts but hyperactive, or normal activity but too much of it. The protein is overproduced. Now, all of these only require one hit. So you get a mutation in one copy and you have the cellular problems here. The loss of function mutations in the tumor suppressor genes require you to have a problem with both of the chromosomes. So here we're looking at somebody who was born with one of these mutations in the maternal chromosome. As long as the paternal chromosome stays normal, it's fine. But then what could possibly happen to this paternal chromosome? Well, first of all, you could lose it. You could have a error in mitosis that causes the complete loss of the chromosome. We could get a deletion that is going to delete the area of the paternal chromosome containing the gene, which means we don't have a functioning copy. We could get a spontaneous mutation in the paternal gene, which is going to have the same effect of we no longer have a functioning copy. Or we could get epigenetic changes that actually silence the normal gene. So things like DNA methylation and histone modification, interfering with your microRNAs, things like that. That is not actually a problem with the gene, but the gene is being silenced and is no longer functioning. So in essence, you don't have a functioning copy of the gene. Doesn't really matter what the cause, whether we're dealing with a gain of function proto-oncogene issue or a loss of function tumor suppressor gene issue. There's only a few different pathways that lead to cancer. The first would be an alteration in cell proliferation. So it is literally growing out of control. It is going through the cell cycle and you have way too many cells. Second would be an alteration in DNA damage response. The cell is damaged, but you're incapable of stopping the cell cycle, so it continues to grow and pass on the damage continually through the generations. Third would be alterations in the growth patterns. So you are seeing an abnormal growth of the cells, meaning that you aren't able to either control the cell cycle, or the cell cycle is not, the growth cycle is not responding to the signals the way that it should. It all leads to cancer, though. Doesn't matter how you're getting there. Okay, so we're going to use hereditary colorectal cancer, which affects the tumor suppressor gene APC. This is actually one of the hereditary cancers, and it is going to be inherited in this way, where you have one non-functioning mutated copy and one good copy at birth. Now, it doesn't mean that you can't develop colorectal cancer in absence of that inheritance. You could absolutely have a spontaneous mutation that would lead to the loss of a copy here, or I'm sorry, both copies of the gene. And you could also have somebody who does inherit this mutation but does not develop colorectal cancer. So to be clear, when we talk about a inherited germline type of cancer, it's not a guarantee. Just like everybody who inherits a mutated copy of BRCA1 or BRCA2 is not necessarily going to get breast cancer. Because remember that cancer is not a one mutation disease. It requires a number of them. Inheriting one of these mutations simply means that you are predisposed to this. So the gene APC is involved in what's called the Wnt cell-to-cell signaling pathway. So Wnt is going to bind a G protein coupled receptor, and when it binds Wnt, you're going to get an active signaling protein. The active signaling protein is going to turn off the APC containing complex. So Wnt inactivates APC. APC is normally degrading beta-catenin. If we enable it, okay, if we inactivate it using Wnt, it means that beta-catenin is going to be stable, and it's going to activate the transcription of your Wnt responsive genes, which leads to an overproliferation of the stem cells in the gut. So when Wnt is able to signal, you're going to get this abnormal proliferation. What APC normally does is it completely degrades beta-catenin. So if you don't have this, you can't activate your transcription complex here. You can't activate these genes. So the normal function of the APC gene is to inhibit the Wnt signaling pathway. If you lose the tumor suppressor gene of the APC, then what you begin to see is excessive epithelial proliferation. You then still have to gain multiple mutations that would lead to cancer. You're going to see this in a video that you're going to watch. So then you would need to have an oncogene activated, usually RAS. You'd have to lose another tumor suppressor. And the final, usually the The last mutation that we would see before we gain the ability to become invasive and metastasize is the loss of P53. So remember that P53 is designed to work at the G1S checkpoint. If you lose P53, whatever mutations are in the cell are going to be copied in the S phase. So this is where we begin to see metastasis and we begin to see cancer. But it took several mutations. It was not simply the loss of the APC gene. We had to lose an oncogene. We had to get another tumor special. We had to continue down this pathway before we actually ended up at cancer. So while cancer is always a genetic disease, it is definitely not just one single mutation. It is multiple mutations. That's why normally when we look at the age that is going to really start seeing cancer be a very high incidence rate, it's going to be really late in life because it takes a long time to get there. It takes quite some time to get there to actually gain enough mutations. Now, does that mean that nobody in their childhood or 20s, 30s, or 40s gets cancer? Obviously not. That absolutely happens. But generally speaking, we tend to see that it takes a long time to go through the processes to gain enough of these mutations that are incapable of being fixed to actually lead to cancer.